Welcome back to the channel. Today we are here to discuss important MCQs on transcription. So let's have the session. Coming to the question number 1, the process of formation of RNA from DNA is known as option A replication, option E DNA repair, option C translation, option D transcription. The correct answer is transcription. Question number 2. Name the site where upstream sequence is located. Option A. Prior to start site or start point. B. After the start point. C. Right border of DNA. D. In the middle of DNA. And correct answer is prior to start point. If we'll consider this is the start point or whatever transcriptional start point, whatever the upstream region will prior to the start point, we'll call it as upstream region. This will call it as a downstream region. In upstream region will contains promoter region, transcriptional binding sites, or whatever enhancers, activators. In downstream region, we'll call it as a gene. The gene which is transcribed into RNA. That we'll look at in downstream region. Question number 3. Which of the following is true for the RNA polymerase activity? Option A. DNA-dependent DNA synthesis. Option B. Direct repair. Option C. DNA-dependent RNA synthesis. Option D. RNA-dependent RNA synthesis. Here the answer is DNA-dependent RNA synthesis. Here, whatever the DNA, from DNA the RNA is synthesized. So it is DNA-dependent RNA synthesis. Next, question number 4. Which of the following ensure stable binding of RNA polymerase at the promoter site? Option A. DNA phosphorylase. Option B. Sigma factor. Option C. DNA glycosylase. Option D. RET-A. And answer is sigma factor. Question number 5. What is the function of the sigma factor in transcription? Option A. Helicase activity. Option B. Transcription initiation. Option C. Transcription elongation. Option D. Transcription termination. And correct answer is transcription initiation. At the start site, so whatever the transcription start site, the sigma factor will bind start site. After this, the transcription initiation will start. Question number 6. Mark the one which is not the transcription inhibitor in eukaryotes. Option A. Rifampicin. Option B. Acridine. Option C. Actinomycin D. Option D. Rho factor. And correct answer is Rho factor. These three are transcription inhibitors. And this Rho factor is transcription terminator. Meaning means here transcription initiated, it stops the transcription itself. Starting of transcription only, but Rho factor it terminates the transcription elongation, terminates the transcription. Coming to the question number 7. Longest primary transcript is created by which gene? Option A. Tintin gene. Option B. Dystrophin gene. Option C. Centromere protein A. Option D. None of the above. And correct answer is dystrophin gene. Question number 8. A DNA sequence is read by an RNA polymerase which creates complementary antiparallel RNA strand is recorded as option A. Secondary transcript. Option B, tertiary transcript. Option C, hexa transcript. Option D, primary transcript. Answer is primary transcript. Question number 9. In eukaryotes, transcription begins only when option A, RNA strand is available. Option B, core promoter sequence is present. Option C, RNA polymerase is available. Option D, none of the above. Answer is core promoter sequence is present. Here, promoter sequence data box is important in promoter sequence. Whenever the data box is there, after the data box, the replication start site will present. Question number 10. The complex of RNA polymerase, DNA template and new RNA transcript is called as option A, transcription bubble. Option B, replication bubble. Option C, translation bubble. Option D, none of the above. And correct answer is transcription bubble. Here, RNA polymerase unit, DNA template, and new transcript of RNA will call it, the entire unit will call it as a transcriptional bubble. Question number 11. Each tRNA molecule has a clover-leaf secondary structure consists of 3 stem loops, 2 stem loops, option C 4 stem loops, option D either A or B. Here in clover-leaf model, the answer is 3 stem loops. Secondary structures are there. This is the one, this is this is this, and this is this. Here codon will be there, and one side anticodon. This is 1 minute. This is the clover-leaf model of the RNA molecule. It has 1 loop, second, third, three stem loop structures will be there. Question number 12. In both eukaryotes and prokaryotes, a promoter region that is rich in AT bases is called as CATT box, B Shine-Dalgarno regions, C data box, and D SV40 region box. And answer is option C data box. Question number 13. Immediately after transcription, which process starts? Option A. Methylated guanine cap is added to the 5' end of the transcript. Option B. Methylated guanine cap is added to the 3' end of the transcript. Option C. Both A and B. Option C. A polyadenylated signal is added. And correct answer is option A. A methylated guanine cap is added to the 5' end of the transcript. This is the mRNA transcript 5' to 3'. After transcription completion, methylated guanine A is added to the 5' end of the cap to prevent the denaturation, to prevent the exonuclease or whatever endonuclease activity, to reduce the endonuclease activity. Question number 14. Transcriptional factors are option A promoters, option B proteins which binds to DNA and regulates the transcription, option C data boxes, option C CAD boxes, and option B is the correct answer. These are the proteins which binds to DNA and regulates the transcription. These are the transcriptional factors. Question number 15. Which of the following statement is true regarding introns? Option A. Introns are the